Now that I know how Bill really feels about physicists, I have to admit I'm a physicist by education and a student of biology. So I thought I'll start off because we are very new to this field, both as an entity, as a company, as well as scientists in this area. I thought I'll give a little bit of a background about the company itself and tell you how we got here. And I want to thank Nancy for inviting me here to present. So who's Chromologic? What do we do? So we have a very well-defined, crisp mission, which is to develop innovative solutions that save lives and make the world secure. So in that mission, for us, we focus in the point of need, or some call point of care, which is a very broad area. It's sort of the point where the care needer, the person who needs care, first interacts with the person who can provide the care. And that could be in the battlefield, it can be at home, or definitely a hospital, mass casualty situations. And so we look at this problem very broadly and come up with solutions. And currently we are developing and patented numerous solutions in this area, starting with a non-contact way of physiologically assessing somebody. It could be from oxidative stress, dehydration, exposure to ionizing radiation. And we do this through an ocular platform where we measure the aqueous humor for biomolecules. So we also go on into areas such as infectious disease. We have developed our own cell lines as hosts for PVivax, so you can do active research. These are currently being tested in Thailand as we speak. Sample preparation is a major part of what we do, mainly because you cannot do point of care or personalized medicine without having a good way to analyze your sample right at the point of collection. We have done years and years of work in sample prep, and we have some products out in this market. And finally, Affordable Care Act. We sit in so many committees, and we see the pain point in healthcare and the transition to a more value-based purchasing. And the hospitals are really not looking for new solutions to solve cancer. They want a cost problem. They're looking for survival. And so we look for ways to help reduce that cost structure through technology. So that's a kind of a big, broad mission, but how do we do this? So we've strongly, truly believe in peer-reviewed research. And we're supported by a strong branch of collaborators. We were founded about 10 years ago. We've built a model of innovation around this structure of being capped and supported on two ends. One is the academia. We collaborate with pretty much every university on the West Coast. And on the east side, we're supported by sponsors, which range from NIH, DARPA, BARDA, and so on and so forth, and really looking at a way of solving these niche societal problems in point of need, which are not being addressed by larger companies. So we founded in 2007, coordinates. We are in the Los Angeles area, and we have a team of about 25 people working full-time, half of us with PhDs in various areas, in physics to biology. So we do have a biologist from our company here. So why are we here? So, you have to be living under a rock not to be doing something synthetic biology if you have such a large mission in mind. So having said that, we actually were getting feedback from our sponsors as well as our collaborators looking at various problems that we work in, from radiation mitigation to cancer to some of the biosensor work that we're doing, that we need to exploit this technology. So we started looking at this problem and said, what can we do? What does a company like us do? So we started looking and said, the largest problem, which is, of course, everybody knows, what is GPRide about, is trying to get large DNA constructs into cells. So I'm not going to teach this great audience here. I've learned more and from what I can probably teach you today, so I won't get into all the details. But essentially, we want to go along this curve. We want to focus on the problem on the far end, very large DNA constructs, and how do you get this into the problem? Into a cell. And to do this, as is pretty apparent to anybody who works in this area, how do you make error-free synthesis of large DNA constructs? How do you manipulate them in the lab if you want to make a high-throughput, low-cost system? Because that's what we do. We want to make this into bend the cost curve so that it becomes a practical solution. And how do you efficiently deliver this DNA into the cell, into the nucleus? Well, this is a big problem. And clearly, everybody has most of the people in this audience have tried this in the lab in many ways as possible. So what does a small company living outside Pasadena come up and do in this area? So we started focusing on was not in things in single DNA, but really at the large end. So that's where our technology comes in, which we call synthetic genetics, or SynGen. And the idea is to meet the unmet need for high yield and quality in linear DNA delivery. So we received some funding from DARPA just last year, and so we're very new to this field. And what we've been building are kits that can be modular and can work in different regions within this whole challenge, starting with. So we are kind of starting at the Gibson method of preparing DNA constructs, but we are happy to take what Bill makes or what or anybody else makes and build that into a modular kit. Second step is how do you purify this? As you know, just handling DNA, large constructs of DNA, what is it? The tensile strength is about 2 millidynes per centimeter squared. Very hard to manipulate large constructs. So we have come up with methods to manage it, and we use a bead-based technology. So our whole process is using beads. So what we have done is optimize this whole construct of beads, starting from the geometry and the chemistry, so that we can approach it through the phagocytotic pathway into the cell. Is that an indication? I must have done something right. Okay. And this has been published before. What we have done is made this into a practical, usable way of, and we are still early. So I think I've got 2 minutes left. I'm going to start wrapping up. Less than 2 minutes left. So what we have done is this is just a snapshot. There's a lot of work that's gone behind this, making microfluidic cell channels to actually process the DNA, really making it efficient. We are right now at about 1 to 2% efficiency in transfection. We've only started working at smaller constructs, 17 kilobase pairs. We're moving up the value chain to looking at these linear DNA constructs. What you can see here is we are kind of solving the basic issues of toxicity, making sure the cells are viable. We can actually, it's hard to see, but we have better pictures hopefully soon. But you can actually see the cells are participating in the mitotic activities in here. And we're using directly human cell lines. I mean, instead of starting in bacteria or viruses, any other form. So we're going directly into human chromosomes. Having said that, I think I will stop here. And what we are looking for are more collaborators. We are new to this field. We'd like to see how we can take some of this technology and make this into a cost-effective platform and into a modular, cost-effective platform. Thank you. There are no questions.
What exactly does the bead offer you in terms of preserving the integrity of the DNA, or is it offering something?
Yeah, it allows us to give us a construct which is large enough that we can wrap the DNA without breaking it, and then that allows us to get it in using the phagocytotic pathway into the cell and release it gently. And we adjust the assays to trigger that process. Thank you.